So the title of our webinar today is Recent Work and Current Methods for Profiling Extracellular microRNA. So the webinar outline will be basically a brief review of microRNA basics, history, biogenesis, and function, and then recent developments of microRNA research in extracellular fluids and exosomes, current methods for sample preservation and extraction of various complex extracellular samples, sample types including exosomes, current methods for microRNA discovery and profiling from extracellular sample types, and finally some case studies and application examples. So before we start, we need to know exactly what is in the circulation or what is in the bodily fluids, different bodily fluids. In the circulation or in the different bodily fluids, there are circulating nucleic acids that are present in different forms. These circulating nucleic acids have many different points of origin. Circulating RNA are found in the circulation as a result of either cell death, by apoptosis, or by necrosis. RNA transcripts released by apoptotic and necrotic cells form complexes with specific RNA binding proteins, or they will be also contained in apoptotic bodies. A subset of these circulating RNA will be also actively secreted from cells in the form of extracellular vesicles, or another name, exosomes. These circulating vesicles contain specific microRNA that are thought to be involved in cell-cell communication. In the last few years, much research has been focused on studying extracellular RNA, with special focus on addressing microRNA expression profiling of different diseases. So next we will briefly talk about microRNA. So microRNA were firstly discovered in 1993. They are single-stranded 21 to 23 nucleotides. They account for approximately 1% of the human genome and are highly conserved in nearly all organisms. It was long speculated that they are just degraded RNA fragments or non-translated small RNAs. MicroRNAs are assumed to regulate more than 50% of all human transcripts. So briefly, we're going to go through the microRNA biogenesis. Most of you know this, but I'll go through it very quickly. Biogenesis of microRNA is a very complex multi-step process. This figure illustrates in details the biogenesis of microRNA. Briefly, microRNA are firstly transcribed in the nucleus by an RNA polymerase II into large precursors called primary microRNA transcripts, which will be then subsequently cleaved into a 70 nucleotide stem loop hairpin microRNA called pre-microRNA. After being exported to the cytoplasm, the hairpin precursors are cleaved, resulting in a small double-stranded RNA duplex that contain the mature microRNA strand and its complementary strand. This mature microRNA strand is selectively loaded into a complex called RNA-induces silencing complex, the RISC. MicroRNAs regulate post-transcriptional gene expression in a sequence-specific manner, where in case of perfect homology between this microRNA and its target messenger RNA, the target messenger RNA will be cleaved by the RISC complex. In case of imperfect binding to partially complementary sequences, especially in the 3' untranslated region of the messenger RNA, the target messenger RNA is regulated by repression of protein translation. So before investigating the microRNA expression in bodily fluids, we need to ask a very important question. Are these cell-free or extracellular microRNA or extracellular RNA stable in these different bodily fluids? The answer is yes. Data have demonstrated that serum cell-free microRNA remained stable after being subjected to severe conditions that would normally degrade most microRNA. Another example in plasma, and despite the presence of high RNAs activity in plasma, incubation of plasma at room temperature for up to 24 hours or being subjected to up to several freeze-thaw cycles had minimal effect on cell-free microRNA levels. Then the interesting question is, why these cell-free microRNAs at these are stable at these harsh conditions? Differential centrifugation and size exclusion chromatography identified two main populations of cell-free microRNA in different bodily fluids, being plasma, serum, urine, or saliva. Most of the cell-free microRNAs were found to be protein-bound, making them inaccessible to RNAs, whereas a minority of specific cell-free microRNA were predominantly associated with vesicles, such as exosomes, which also made them inaccessible to RNAs. Then the next question is, why to study extracellular microRNA? Since microRNA expression was found to be tissue-specific, microRNA signature profile has been partially identified for each tissue. This specificity, together with the remarkable stability of these cell-free microRNA, make them useful biomarkers with robustness and reproducibility in cancer diagnosis. The assessment of these microRNA signatures can accurately predict prognosis in various types of cancer. So in cancer, microRNA was found to play an important role in tumorigenesis and basically in every step of carcinogenesis, including invasion, metastasis, apoptosis, and drug resistance. Depending on their downstream target genes, microRNA can function as a tumor suppressor if it targets an oncogene. Also, microRNA can function as an oncogene if it targets a tumor suppressor gene. I have highlighted here some publications that use different bodily fluids such as serum, plasma, urine, CSF, saliva, breast milk, and cow's milk for the discovery of specific microRNA or microRNA expression-based signature that can act as a potential biomarker for different diseases. Also, this table summarizes some of the differentially expressed microRNA with a diagnostic and prognostic value in patients with different cancer types. Now, you are starting a new project to discover a biomarker for certain disease from a bodily fluid, let's say plasma. Before you start, you need to know what kind of challenges you will be facing and how to overcome such challenges. The number of samples to be included in your study is the first challenge. Data on circulating microRNA are still limited because the number of patients are small. Circulating RNA in plasma or serum, for example, and then the same in other bodily fluids, are normally found in a very low concentration, which makes it very challenging to extract, and usually large sample volume may be required. Since you will be dealing with small RNA with very low RNA concentration, you need to choose the best extraction method that is robust, consistent, and easy to use. Normalization is also very important in the analysis of differential gene expression. Since now, no constitutively expressed circulating microRNA have been found. Therefore, normalization is very tricky when dealing with bodily fluids RNA. No universally implemented guideline for the collection, extraction, normalization, and analysis of sample for microRNA analysis. Therefore, various platforms techniques exist. One of the most important challenges is how to prepare your sample for RNA extraction and which method to use for RNA extraction. So levels of cell-free microRNA expression are known to be significantly affected by pre-analysis factors, such as sample preparation and RNA extraction methods. For the sample preparation part, cell debris, apoptotic bodies, and blood platelets contain considerable amount of microRNA, And contamination of your samples with these microRNA from these sources will significantly influence the final levels of the cell-free microRNA. Preparation of plasma or serum from blood samples is very critical, as some anticoagulants, such as heparin, can inhibit some of the downstream applications, such as reverse transcription, whereas plasma prepared from blood collected on other anticoagulants, such as citrate or EDTA, will not show such an inhibitory effect. Another challenge is the hemolysis of blood during the preparation of your plasma or serum. And hemolysis is known to markedly influence microRNA expression patterns because of a contamination of the abundant intracellular microRNA in these types of sample in your plasma or serum. In other bodily fluids such as urine and saliva, it is also important to remove cells and cell debris completely, as they contain abundant microRNA that may significantly influence the final level of your microRNA. As for the RNA extraction, different and inconsistent microRNA profiles may be obtained depending on the extraction method, which will bias the analysis and will bias your results. Now you have your sample, a bodily fluid, cell-free, and is ready for RNA extraction. And which method you will be choosing for your RNA extraction. For purifying RNA and DNA, we are using methods that have been developed for quite a while. Phenol-based, for example, which is very common. Secondly, column-based, using silica membrane. These methods have served very well, particularly for samples input that it was originally designed for, such as cells or tissue with high RNA amounts. As a natural transition, people have started to fit these types of extraction methods to isolate RNA from liquid biopsies, such as plasma, serum, urine, or even exosomes. The question is this suitable? Bear in mind that while these methods are good for certain types of samples, they still have some shortcomings, especially when dealing with samples with very low RNA inputs. One of these shortcomings, for example, is the loss of diversity, especially when extracting RNA using phenochloroform. In addition to the inhibitory effect that is present in the RNA extracted using phenochloroform on microarrays and sequencing, phenochloroform extraction procedures tend to have a bias in RNA recovery, particularly microRNA, based on the GC content, and therefore not all the microRNA profile will be identified. Some microRNA may be thought to be differentially expressed, but actually, due to this bias in the extraction method, they were not captured from the beginning. Another shortcoming is the size bias when using silica-based column, and also the inconsistency in the isolation. Silica column sample prep has size bias, leading to the loss of important small regularity RNA below 200 nucleotides. And as it's shown in front of you with the silica column, beautiful 28S and 18S ribosomal RNA, very intact RNA, but the most important RNA species that we will be discussing today is the small RNA, and it's completely missing or lots of it is missing. So this is very important. Imagine you are isolating RNA from plasma and you mainly are isolating these small RNA and you can't find it. New form of biological inputs such as extracellular RNA has new requirements of RNA isolation. Low quantity per volume, in our case from plasma, serum, urine, it has very low amount of RNA. Also, the small size of the RNA species that we are isolating. So for provider for various RNA extraction kits, they have to modify the existing protocol to fit into the requirement. Whereas to capture low RNA inputs, for example from plasma, also with a higher consistency. This table shows that some extraction methods are now combining phenochloroform for the isolation of small microRNA, followed by a cleaning step on a silica-based column to remove any inhibitors. Still, this is time-consuming, using hazardous material like phenol, as well as it's not going to be cost-effective anymore. Also, there is a bias in the RNA recovery based on the GC content, as we discussed before. As for the case of cell-free RNA, there is either a combination of these classical methods with additional supplements such as carrier RNA. And the use of carrier RNA is to overcome the consistency issue. And these methods were mainly designed for high RNA inputs and not for small RNA inputs that are normally found in such bodily fluids. So Norgen has a unique platform technology based on a proprietary resin matrix, which is silicon carbide, that overcomes some of the shortcomings of both the phenol-based method, which is loss of diversity, and also silica-based extraction methods, which is the loss of important small regulatory RNA, especially below 200 nucleotides. What we found that silicon carbide can bind to all types of DNA or RNA, including small RNA and microRNA, without the use of phenol, and therefore the RNA isolated will be inhibitor-free, which is going to be suitable for lots of downstream applications, sensitive downstream applications like RT-qPCR, microarray, and next-gen sequencing. Also, silicon carbide has an outstanding sensitivity, reaching the single-cell level, as well as can capture low RNA concentrations from bodily fluids that are normally have very low RNA concentrations without the use of carrier RNA, because also carrier RNA may interfere with the normalization procedure. So the use of silicon carbide also in a slurry format allows it to accommodate large sample volumes. Since the RNA in such samples are very low, so we may need to increase the sample input volume in order to recover more RNA. This is going to be very difficult when using phenochloroform or the regular silica-based columns, but using silicon carbide in a slurry format allows the silicon carbide to accommodate large sample volumes for the extraction of RNA normally present in low amounts in liquid biopsies or bodily fluids. This slide showed that silicon carbide was able to bind RNA sequences at equal rate without bias and without the use of phenol, as opposed to a phenol-chloroform method that is coupled with a silica-based matrix. Most importantly, all RNA sizes, especially small RNA below 200 nucleotide, was also extracted. Therefore, we are not missing anything for our downstream application or microRNA expression profiling. This is shown also by the high number of different microRNA that was detected from only 100 microliter plasma using Norgen total RNA kit, compared to the small amount of microRNA recovered from 600 microliter plasma using miRNA Paris, which is a combination of phenol-chloroform and silicon. And again, because of the phenol-chloroform, there is a bias in the GC content. The silica-based, there is loss of the size. Therefore, the silicon carbide obviously was able to recover more microRNA because of these advantages. So while diversity is important, it is intimately linked to consistency. For good RNA extraction, it is desirable to have both diversity and consistency. This is especially important for precious clinical samples, where you have only one shot to study. Hence, you want to isolate RNA, and there is no doubt that RNA extraction works. As this slide shows, the combination approach for isolating total RNA large and small, used by other companies, that is combination between phenol and silica based, showed high variation in messenger RNA recovery. Also, extremely high alcohol concentration to bind RNA to column, which causes precipitate out effect. So this inconsistency is very profound when using these methods. So now for the ease of use, these are the different isolation methods, being phenol or silica, or phenol combined with silica and silicon carbide. The slide showed the advantage of Norgen's simple approach for RNA extraction. While the difference may look like 5 to 10 minutes, it may do so when the samples number are very low, 1 to 6 samples. But imagine this is hundreds of samples, then the difference could be very dramatic. So this is a very important slide showing the difference of the ease of use using Norgen's silicon carbide approach to extract RNA when dealing with large-scale cancer biomarker screening. This is a large-scale NIH cancer biomarker screening involving serum obtained from 410 individuals. The consistency was very good. Imagine that you are using a phenochloroform or phenol silica kit. This is going to be an equivalent study, will require 410 phenochloroform, which is going to be very problematic. So now, after extracting your extracellular RNA from your sample, what next? You will need to measure the RNA concentration and assess the quality of your RNA for either RT-qPCR for gene expression analysis, microarray, or sequencing. One of the main problems associated with circulating microRNA extraction and comparison of sample collectives is the quantification of microRNA. In contrast to isolated messenger RNA, where the size and relative abundance of ribosomal RNA can be used to assess the integrity of the RNA preparation, there is currently no good way to assess the quality of isolated microRNA. In addition, degraded messenger RNA can also interfere with the quantification of microRNA preparation. The low abundance of microRNA in serum and in different other bodily fluids can hardly be determined using spectrophotometers, nanodrop, bioanalyzer. A robust and sensitive methods for the analysis of body fluid microRNA is their relative quantification by stem loop reverse transcription RT-PCR for one of the most abundant microRNA, which is miR-21, for example, or you can use a small RNA, housekeeping RNA transcript like the 5S ribosomal RNA. This has been widely used for the sensitive detection of low abundance circulating microRNA with high reproducibility. So the amount of cell-free RNA significantly varies between sample to sample. That's what you should be expecting from RNA isolated from bodily fluids. Typical yields of free circulating and exosomal RNA vary depending on the input sample, as the amount of RNA present in bodily fluids will depend upon the health status of the individual. Normally, RNA yield from bodily fluids is highly variable, where this variability is also observed between samples collected from the same donor at different times during the day. Another measuring stick is the 8:60:80 to assess the quality. 8:60:80 ratio tend to be significantly affected by the concentration, and hence RNA concentration in such bodily fluids is normally low. Therefore, you should be expecting a very low 8:60:80. This 8:60:80 will not affect any of your downstream application, as it's not really representing the quality, but because the RNA concentration is very low. Since these bodily fluids are cell-free, then do not expect to see any ribosomal RNA like the 28S or 28 or 18S on either a bioanalyzer or agarogel. If it happened and you observed that these 28S and 18S on a bioanalyzer trace or on an agarogel, this indicates that your bodily fluid during the preparation was contaminated with cellular RNA. This will bias your cell-free microRNA expression analysis. Since the ribosomal RNA will not be showing, and since the majority of the RNA in such bodily fluids are short RNA fragments, then expect also to see a low RIM value. The bottom line here is do not use these methods for assessing the quality and quantity of RNA isolated from cell-free bodily fluids, as they will give you unreliable numbers because of the low RNA concentration, and normally these RNA concentration are below the detection and the sensitivity levels of such methods. As mentioned earlier, the only reliable method to assess the quality and the quantity of cell-free RNA is RT-qPCR against one of the small housekeeping genes like the 5S ribosomal RNA and U6 that can also be used for normalization. Also, the stem loop RT-qPCR amplification of MER21, which is highly abundant in most of these bodily fluids. Christoph?
So once you have the your microRNA isolated, and typically the yields are pretty low from this type of samples. So typically, no matter what type of follow-up experiment you're doing with the actual sample, you will have to amplify your microRNA. So I will go into the different types of experiments that you can do with your microRNA samples, and focus there specifically on, of course, arrays and next-gen sequencing, and then highlight a couple of customer publications. So just to give you a brief overview about the different types of different steps in microRNA research. First of all, of course, you have to identify your microRNA. So there are different options: genetic screening, direct cloning, and then sequencing. Nowadays, there are a lot of computational strategies, and really lots of different databases also online. Here are a couple of them listed. In the early days, a lot of predicted microRNAs were verified using tiling microarrays. I remember specifically the miRNA 7 update. I mean, this is a couple of years back. What was done on our microarrays, and then of course that was kind of replaced by next-gen sequencing for the discovery step. If you look at the detection and profiling step, of course the classic method, Northern blotting, then in situ hybridization, basically arrays. Real-time PCR, of course, is the gold standard when it comes to quantification of specific microRNAs. This is typically used by our customers to validate microarray or next-gen sequencing results. Basically, once you've done the differential profiling and you know which microRNAs you want to focus on, then typically PCR is a good method. Then microarray analysis for the profiling, overall profiling of your samples, and as well as next-gen sequencing. And as mentioned, I will go into detail for these both methods. Then the big step, of course, the big issue is the target determination, especially because you don't have a perfect match to the messenger RNA in mammalian samples. In plant samples, it's a little easier because you do have a perfect match. Again, there are a lot of different databases out there nowadays that predict the targets that are available online. Here, just a few highlighted: TargetScan, PICTAR, and PETA. If you look at the experimental, on the experimental side, you have gene and proteome expression analysis, then pull-down assays, 5' erase analysis, and degradome sequencing, which again is particularly helpful for plant samples, but has been used also for mammalian samples. Once you know the target, of course the pathway analysis is of interest, and there are a couple of free tools available that are listed here. For example, miRFocus. Most of them focus on the human, or maybe sometimes also the mouse genome. There's of course also commercially available software solutions are there, like Ingenuity, to mention one, and probably the biggest. Then when you look at the functional analysis, typically luciferase assays are being done. Then of course gene knockouts or overexpression models. Using antagomers you can do microRNA inhibition studies or microRNA mimicry studies. Then when it comes to databases, I just want to highlight two as they are particularly interesting here for this webinar: miRNA and HMDD. miRNA I will feature briefly on the next slide, but HMDD is basically a database that relates human microRNAs and to their diseases, and stands for Human microRNA Disease Database. So and HMDD was just updated in January of this year, so it's pretty recent. It doesn't include the latest miR-based version yet, but it's still very up to date. As mentioned, miRNA is probably of particular interest here to this audience today because it's the extracellular circulating microRNA database. And basically what the slide is showing is that it interfaces with a lot of the other databases, and particularly miRO, which gives you associates the microRNAs with the phenotype in human. And of course also miR-based, basically the mother of all microRNA databases. So this just as a reference for you, and of course the link to the article. As Chris mentioned already, all the links will be clickable in the actual PDF file that you will receive, so you don't have to write down the references. When you're starting out, you probably have a particular sample type in your hands, and you have a question that you want to solve. And there are the different methods out there: qPCR, microarrays, and next-gen sequencing, or miRNA-seq or rna-seq. I found this particular review paper very interesting because it gives you a good guideline on which method to choose to answer a particular question that you might have, because depending on the sample type and the question that you're trying to answer, you might want to use a different method. For example, typically if you want to do overall profiling, microarrays are still a very good method, and another option for the overall profiling is next-gen sequencing. And on the other hand, if you're looking really just at a subset of microRNAs or just very individual ones, then probably qPCR is the more cost-effective method to choose. So this just as a recommendation has a very nice flowchart and really good paper. It's from 2012, May in Nature Reviews Genetics. As mentioned, I want to focus on microarrays and RNA-seq solutions here, and compare them and what to pick, and advantages and disadvantages. So if we look at the microarrays, it's of course a robust method. Had initially a bad rap due to the early years in the gene expression field, but once it hit the microRNA community, it was already proven and reliable. So it's a very reliable, high throughput method. Can be easily automated. The data analysis is pretty straightforward. It's low cost. You're profiling the all known microRNAs, and the turnaround time is typically very short. If you do it yourself, of course it's up to you, but if you use a service provider, it's typically between 2 and 3 weeks, depending on the number of samples. We also, in our case, we also offer a rush service for 5 business days. So that was the the method that the microRNA community started out with for profiling, and then especially for the discovery, next-gen sequencing took over. So you will find different terms: RNA-seq, microRNA-seq. It's pretty much all the same. Basically, next-gen sequencing using RNA samples. It has the advantage that it can give you a much broader view, comprehensive view of the transcriptome. So you can run samples not only to look at the microRNA level, but also, of course, messenger RNA, non-coding RNA, long non-coding RNA, etc. And a big advantage is, especially when you work with species that are less well characterized, is that you don't need any prior sequence knowledge. That's basically the limitation for the microarrays. You need to know the sequences so that you can put the corresponding probes on the chip. So besides that, of course, you have a dynamic range and sensitivity that's somewhat tunable in next-gen sequencing. And you can also detect structural variations like single point mutations, gene fusions, alternative splicing events, and so on. And of course, it's a truly digital solution, meaning it's a quantitative method. That said, you have to there's the caveat that for microarrays you're using your microRNA or your total RNA sample directly, versus next-gen sequencing approaches where you have a pretty elaborate sample library prep involved. So there's still a lot of discussion going on in to which extent you're actually biasing your profile in the library step phase of next-gen sequencing. And there was just recently, 2 or 3 weeks ago, a paper that showed that you're actually biasing your profile there, and it's really not truly quantitative. So let's focus on the microRNA just briefly. Typical steps involved is, of course, you isolate your sample. Then this is basically our workflow here in-house. First thing, of course, you do a sample QC. Then you prepare the sample for the array. You run the hybridization reaction, label it, do the data extraction, processing, normalization, and so on, and do then the analysis. And what you get at this point is basically a list of differentially regulated microRNAs. And typically, once you publish it, you're required to submit the data to GEO or a similar database where they are publicly available. And of course, we help our customers with that as well. Then, as mentioned, you need to know the microRNAs in the case of the microarrays, and so it's important to have always the latest miR-based version probe content on your chips. So there are chip manufacturers, different chip manufacturers out there, or different services out there. Just pay attention to the miR-based version because there can be quite some differences. So in our case, we always are in sync with miR-based. Basically, as soon as miR-based, the publicly available database, updates their probe content. Right now we're at version 21. We basically update it the same day. So right now for human, for example, there are 2,565 mature human microRNAs that are known. Typical microarray experiment, of course, is that you run a couple of biological repeats. We will run them on individual chips. Then you do some normalization, clustering analysis, and what's shown here is basically just the overall view heat map of control versus treated, and then you can do statistical tests like simple student's t-test to differentiate, to reveal the differential regulation between two different sample groups, in this case control and treated, and then you get results such as those here on the right at different p-values, which stands for the statistical significant significance of the results, and yeah, from there on you go on to look at the potential targets, pathway analysis, etc. As mentioned, rna-seq kind of took over at least the discovery step, but a lot of customers use it also for the profiling step. It really depends also on your budget, quite frankly. So let's focus on this for now. So the workflow is pretty similar. You have the sample QC, of course, initially. The big difference is, as mentioned before, you have a library that you need to prepare so that you can run it on the sequencer. And at this step, you typically barcode the samples because, as I will mention in the following, you typically run multiple samples in one sequence, so-called sequencing lane. Then you do the actual sequencing and then the data analysis. And one thing that comes up on a regular basis that we notice is that is the number of biological repeats that you need to run in your experiment. For some reason there seems to be the preconceived notion that RNA-seq is the new magic bullet, and we encounter this quite frequently, that people think, oh well, this method is so awesome we don't need any biological repeats any longer. And of course that's not the case. So this is just as a reminder that experimental design, no matter which method you're choosing, is really crucial. You need multiple biological repeat if you want to have any statistically significant data. So if you just send us a cancer patient sample and a control patient sample, then the difference, you will see a difference probably. But of course you cannot draw any meaningful conclusions based on that. Because the variation that you see might be just simple patient-to-patient variation. There's no indication that this difference would be caused by cancer in this example. So the number of biological repeats depends on the type of samples you have. Typically, when you have just regular cell line samples or tissue samples, we would recommend the very minimum of 3. Of course, 3 to 5, let's say. That will give you some good initial data. That's the 3 to 5 samples per sample group. And then when you look at actual patient samples, then you have to increase the number by quite significantly, typically at the minimum 10 samples per group, sometimes even up to 20, 30, and so on. Then let's just look at the actual sequencing step. So we're using an Illumina HiSeq 2500 machine. The Illumina sequencing is pretty much the de facto standard in this field nowadays. Our read lengths are 50 bases. Number of reads are roughly 100 million per lane. You can tune, of course, the machine depending on whether you want to focus on the depth or the speed. Basically the throughput. So that's why you will probably oftentimes see different numbers for the same machine. We typically recommend up to 10 samples per lane. That gives you roughly between 7 to 10 million reads. Ideally, it depends, of course, also on the sample quality. And basically, it's kind of up to you what type of where you draw the line between depth of sequencing, meaning the number of reads per sample, and the cost. So if a lane has 100 million reads per lane and you put only one sample on here, that's shown here in this table, you get, of course, 100 million reads per sample. And as you increase the number of samples, of course, the number of reads per sample gets reduced. So that's where a couple of years ago, I remember being at a conference and somebody mentioned the number of $25 per sample. And of course that's true if you put tons of samples per lane, but you don't get any meaningful data. So you want to have a minimum number of reads per sample, and that's why we draw typically the limit at 10 samples per lane for microRNA or small RNA sequencing. That differs, of course, for total RNA or transcriptome RNA analysis. This is just a brief overview of the workflow that our software does, or that we go through when analyzing the data. Basically, you have raw reads, you map them, transfer them into mappable reads to the genome, and then you have basically two groups: those that do match mammalian microRNAs and miR-base, and those that don't. This is assuming a mammalian sample, of course. And then you basically go through different mapping processes and categorize the samples. And at the end of the day, you're revealing potential new microRNA sequences that you then can validate using qPCR. These are basically the two big methods that are standard nowadays and that we offer as a service. And in the following, I just want to highlight a few customer publications briefly. Some of them also use the Norgen Biotek isolation kits, and basically go through a couple of case studies, basically to give you an idea of what has been done and what you could do. So the first one is focusing on the urinary exosome. It's a publication from last year from Gilles et al. And it was focusing on the salt sensitivity of blood pressure, basically looking at the kidney cell-derived microRNA and whether it can be used as an extracellular biomarker. And so that is important because salt sensitivity of blood pressure is linked to a higher incidence rate of cardiovascular disease. And the method that was used was microarrays. So basically, human urinary exosomal microRNA samples were sent to us, and we ran the arrays for them. This was done with miR-base version 18 back then, so we had just under 1,900 different microRNAs that were known at the time. And three different groups were profiled. You have the saline-resistant, labeled here as SR group, the saline-sensitive ones, labeled with SS, and then you have the inverse sensitive salt-sensitive group here, ISS, here to the right at the bottom. And so a total of 45 exosomal microRNAs were identified to be differentially regulated, depending on which case. So you have 11 that were upregulated in this case, and then 13 down here in the comparison between the resistant and the sensitive case. And then you have another 21 that were identified for the inversely related sensitive ones. So that's a pretty common approach, basically, to first profile and see which microRNAs can be used as a biomarker, as a potential biomarker. The next case features a sequencing application using, in this case, it was done using pig breast milk exosomal microRNA. And basically this is a study across the entire lactation process using Illumina sequencing. And what they found is that immune-related microRNAs are enriched in the breast milk exosome. And what was of particular interest is that the exosomal microRNAs that are related to immunology are increased, or particularly increased in the colostrum versus the mature milk. So the colostrum is basically the pre-birth milk, and it's interesting because apparently this microRNA information gets transferred into the newborn, and it gets transferred probably through the digestive tract. And it's interesting because initially when studies about breast milk came out, it was people really questioned these results because it seems odd that they're that stable, but it has been shown that microRNAs actually can be very stable under even harsh conditions. Oh, and just one point here, this is basically the graphic is showing here the difference between the colostrum-only fed population and the mature milk-only fed population, and then looking at the different microRNA levels for the microRNAs that are important for the immune system. Then, if we're looking at an overall view, an experiment that takes an overall view at the extracellular vesicles, this paper comes to mind from Biotek et al. Published this year. It's basically the characterization of the entire small RNA content of trypanosoma and cruzi, which is responsible for the so-called Chagas disease, which is predominantly prevalent in Central and South America. So it's a genome-wide analysis of the trypanosoma-derived extracellular vesicles, and they looked at the infective, non-infective in blue and versus infective stage of the parasite, and looked at the different RNA content. And it's interesting because it reveals that the variety and expression of the small RNAs are actually different between the different parasite stages, suggesting different function. And so this is shown here on the right. You have, again, the blue non-infective versus red infective stage, and then a different looked at the different type of RNAs, ribosomal RNA, tRNA, and so on. And you see there's a clear difference in overall RNA profile. And yeah, bottom line of this paper is basically it calls attention to the potential regulatory functions of these RNAs and that they might play once they're being transferred into the host, or also in the parasite itself, of course. So again, that was done with sequencing, and is an overall view at the transcriptome level. Then let's switch to the more common sample types, serum and plasma. Here again, a sequencing experiment looking at different filarial parasites, meaning worms, that are animals or humans are typically infected with. And what was done here is basically they looked at they identified in the study a total of 200 mature microRNAs of nematodes in the serum and plasma samples. They looked both at human as well as canine samples, and they identified 200 mature microRNAs of nematodes that originated from nematodes in the plasma of heartworm-infected dogs. And on top of that, they also identified 21 microRNA candidates that were or are predicted to be released from nematodes that causes the river blindness in human. And based on those results, they then developed a new, basically, diagnostic method for heartworm disease in dogs based on microRNA 71 and microRNA 34. And I don't know how cost-effective that is, but if you have a dog, especially here in the southern region of the United States, and you have to get a yearly heartworm test, and you typically have to wait a couple of days for the results. So with a simple qPCR, that can be certainly be more time effective. So another sequencing sample study by Tan et al. It's using was a study of serum, human serum, and looking at the comparing regular healthy individuals versus those that are suffering from NAFLD, which is non-alcoholic fatty liver disease. And the result of the overall profiling using sequencing was that you can use 4 specific microRNAs that are listed here to diagnose the disease, and with a very high sensitivity and very high specificity. Sensitivity over 90% and specificity around 76%. And what this graph is showing basically are the levels of microRNA, the microRNA expression levels in control versus disease patients. And you see the first 4 here were included, microRNAs were included in the final diagnostic panel. The 2 samples here on the very right and shown in the graph were not included. So this is basically a typical application, again, using serum samples, finding a biomarker for a disease, and potentially be able to detect diseases very early on before it reaches the phenotype level or the proteome level. Okay, then another big application, of course, besides cancer and heart disease is diabetes. A lot of customers work on this, and this is an experiment that used next-gen sequencing and then followed up with qPCR. Again, the profiling step initially was the next-gen sequencing, and then looking at a subset using qPCR, and the qPCR results are shown here in the table. And to make it short, there were 8 microRNAs found to have a significantly lower expression levels in type 2 diabetes patients, and especially microRNA 23A, that's why it's highlighted here, is a potentially very valuable biomarker for early detection of type 2 diabetes with a very high sensitivity of 79% and specificity of 75%. And one final paper looking at cancer, again using serum plasma sample. It's looking at pancreatic cancer, which is a really important field because it has the poorest overall survival rate among human cancers, and there's also the absence of screening tools available right now that contribute to that. So this was a microarray study and was already published in 2010. And if you would do it nowadays with more known microRNAs, probably you would get even more microRNAs that you would be able to use as a diagnostic marker. But basically, microRNA 21 and LED7 were identified as playing an important role. The increased levels of microRNA was correlated to basically a bad outlook, worse survival rates for the patients, and a downregulation of LED7 was indicating the same thing. And important to note was also that the microRNA 21 family was overexpressed in chemo-resistant pancreatic cancer cells. So it's not only in this case a diagnostic marker, but also gives you a hint of how to potentially treat, how to pick the treatment for a patient. So this just is a brief overview and some application examples. Thank you for joining today. I hope you got some valuable information,